For as long as the discipline of genetics has existed as a scientific enterprise capable of reading the molecular record of human ancestry, one of the most persistently compelling and persistently contested applications of its methods has been the effort to trace the biological threads that connect living Jewish populations to their most ancient predecessors. Not through sacred text, not through rabbinic genealogy, not through the oral and written transmission of historical memory, but through the actual physical evidence preserved inside the nuclei of human cells across thousands of years of diaspora, dispersal, persecution, migration, and survival. That effort has produced, over the past 2 decades, findings of extraordinary depth and precision that have fundamentally transformed what is possible to know about Jewish genetic history. And some of the most significant of those findings have been, in the most technically precise and scientifically honest sense of the word, completely unexpected. Not unexpected in the sense of being implausible in retrospect, once the evidence has been examined and the historical context has been understood. Most great discoveries look inevitable after the fact, when all the pieces are visible and the picture they form has become familiar enough to feel obvious. Unexpected in the deeper sense that no existing model of Jewish genetic history, no prevailing academic theory about the origins and migrations of Jewish populations, and no interpretation of the available documentary and archaeological evidence had predicted them before the molecular evidence arrived. The ancient DNA revolution, the set of technological advances that has made it possible, since approximately 2010, and with rapidly accelerating power and resolution since then, to extract, sequence, and analyze the genetic material of people who lived and died hundreds or thousands of years ago, has given the study of Jewish origins a tool of extraordinary power. And that tool has been revealing connections that the history books had either missed entirely or actively contradicted. To understand what those connections are and why they matter, not just to scholars of Jewish history, but to anyone who thinks seriously about the relationship between genetic evidence and the stories that human civilizations tell about themselves, it is necessary to begin with the specific and remarkable circumstances in which ancient Israelite DNA was first successfully recovered, analyzed, and compared against the genomes of populations that no one had previously thought to place in the same genetic lineage. That story begins in 2018, in a village near Jerusalem, in a tomb that had been broken open by thieves before it could be properly excavated, and it unfolds from there into a network of connections spanning continents, centuries, and civilizations that the people buried in that tomb could never have imagined being part of. In 2018, the Theft Prevention Unit of the Israel Antiquities Authority discovered a tomb in the village of Abu Ghosh, located immediately adjacent to the biblical settlement of Kiryat Yearim, approximately 15 kilometers west of Jerusalem. The tomb had been badly damaged before its discovery, its original contents partially disturbed, its physical integrity compromised in ways that made systematic excavation both urgent and methodologically challenging. But what the tomb contained, despite the damage, was something that researchers in the field of ancient DNA had been seeking for decades without success. Human remains from the Iron Age, from the period of the First Temple, from the era in which the people we recognize as the ancient Israelites were living and building their civilization and transmitting the religious and cultural traditions that would eventually become the foundation of 3 of the world's major religions. For the first time, ancient DNA was successfully recovered from the bodies of ancient Israelites living in the First Temple period. This achievement, described by experts in the field as a genuine milestone in the study of lost civilizations, was enabled by the specific preservation conditions inside the Kiryat Yearim tomb and by the application of analytical methods refined to the point where usable genetic data can now be extracted from ancient bones that would have yielded nothing to the techniques available even a decade earlier. The researcher leading the archaeological investigation at Kiryat Yearim was Israel Finkelstein, an archaeologist at Tel Aviv University and the University of Haifa, one of the most respected figures in the field of Levantine archaeology, and a scholar whose careful, evidence-driven approach to the biblical period had already made him one of the most influential and occasionally one of the most controversial voices in discussions of ancient Israelite history. I suppose that we are dealing with a family tomb, likely from the elite of Kiryat Yearim, whose members were buried there over a long period of time, Finkelstein noted in early reporting on the discovery. The significance of the location added another layer of historical resonance to a find already remarkable enough in its scientific dimensions. Kiryat Yearim is mentioned in the biblical text as the settlement that housed the Ark of the Covenant before it was brought to Jerusalem, a detail that places the site at the very center of the biblical narrative of the early Israelite monarchy, in the period of transition between the age of the Judges and the establishment of the Davidic kingdom. Whether or not the biblical account of the Ark's history is taken as literal history, the reference establishes Kiryat Yearim as a location of significance in the earliest layers of the Israelite tradition, which made the recovery of genetic material from a First Temple period tomb at that precise location an event of dimensions that extended well beyond the purely technical achievement of ancient DNA extraction. The genetic data from Kiryat Yearim offered, in the words of the researchers involved, a first glimpse of how directly sampling remains from the First Temple period can shed light on the history of the ancient Hebrews, Israelite or Judahite. This careful phrasing reflects both the genuine scientific significance of the find and the appropriate epistemic humility of researchers, who understood that two individuals from a single tomb represent a preliminary result, not a definitive characterization of an entire ancient population. The preliminary results were consequential enough, however, to establish that the Iron Age population at Kiryat Yearim displayed a genetic profile with strong continuity to earlier Levantine populations, a finding consistent with the broader picture that the ancient DNA revolution had been constructing piece by piece across the entire region. But it was when this First Temple period data was placed in conversation with the results emerging from a completely different direction of research, the investigation of a tiny, isolated, historically marginal community living today in a suburb of Tel Aviv, that the truly unexpected connection began to come into focus. The Samaritans are one of the most extraordinary survivals in the entire history of the ancient Near East. They are a community of approximately 750 people, about half of whom live in Holon, near Tel Aviv, and the other half near Nablus in the West Bank, who have maintained their religious and cultural identity continuously since antiquity, and who have always claimed descent from the ancient Israelite tribes, specifically from the northern kingdom of Israel, which was conquered by the Assyrian Empire in 721 BCE, and whose population was supposedly dispersed so completely that 10 of the original 12 tribes of Israel simply vanished from history, becoming the legendary 10 lost tribes whose fate has generated speculation, mythology, and occasional serious historical inquiry ever since. The Samaritans have never accepted this disappearance narrative. They have insisted, across more than 2.5 millennia of history in which they have been marginalized, persecuted, forcibly converted, and reduced from a population that numbered more than 1 million in late Roman times to fewer than 150 individuals in 1917, that they are the authentic heirs of the ancient Israelite tradition, more authentic in their own self-understanding than the Jews who rejected their legitimacy. The mainstream Jewish tradition had, for most of recorded history, treated this claim with deep skepticism. The biblical texts themselves present the Samaritans as a mixed population introduced into the former northern kingdom by the Assyrians after the deportation of the original Israelite inhabitants, a population of foreign settlers who adopted a syncretistic version of Israelite religion without genuine genealogical or spiritual connection to the original covenant people. This portrayal had shaped Jewish attitudes toward Samaritan claims of shared ancestry for centuries, and the Samaritans had largely been treated as a people whose insistence on their Israelite heritage was a form of historical fabrication, motivated by a desire for religious legitimacy they had never genuinely possessed. Then the genetic evidence arrived, and it said something fundamentally different. A landmark genetic study revealed a close connection between Samaritans and Jewish Kohanim, the priestly class within Judaism, who traced their lineage to Aaron, the brother of Moses, and whose special ritual status within Jewish law depends entirely on the maintenance of an unbroken patrilineal descent from that ancient priestly ancestor. The Y chromosome, the portion of the genome transmitted exclusively from father to son, preserving the patrilineal line intact across generations in the same way that Jewish tradition preserves the Kohanic identity, showed that Samaritans and Jewish Kohanim shared genetic ancestry that predated the Assyrian conquest. The Y chromosome clearly shows that the Samaritans and the Jews share common ancestry dating to at least 2,500 years ago, according to the study's authors. A community that mainstream Jewish tradition had characterized as genealogical imposters turned out, when the molecular evidence was examined, to share deep and specific biological kinship with the most genealogically prestigious class within Judaism itself. The implications of this finding radiated outward in multiple directions simultaneously. For the Samaritans, it represented a scientific validation of claims they had been making against persistent rejection for more than 2 millennia. For historians and geneticists studying the 10 lost tribes, it suggested that the disappearance of the northern Israelite population was considerably more complex and considerably less complete than the traditional narrative had assumed, that some portion of the population that the Assyrian conquest supposedly eliminated had in fact remained in place, intermarried, adapted, and persisted in a form that retained enough genetic coherence to be identifiable 2,500 years later. For the broader study of Jewish genetics, it opened a window onto a population that had been almost entirely neglected in previous research programs focused on the major Ashkenazi, Sephardic, and Mizrahi communities, and that turned out to carry genetic information of genuine historical significance, precisely because its isolation had preserved genetic signatures that the larger and more mixed Jewish communities had diluted beyond easy recovery. The unexpected nature of the Samaritan connection was remarkable enough on its own terms. But the ancient DNA research program that has been transforming the field across the past decade has revealed another lost connection of a completely different kind. One that reaches not across the religious and political divisions of the ancient Levant, but across the full geographic breadth of the Eurasian continent, connecting the genetic heritage of one of the most studied Jewish populations in the world to a civilization that most scholars of Jewish history had never thought to include in the story at all. Contemporary Jews retain a genetic imprint from their Near Eastern ancestry, but obtained substantial genetic components from their neighboring populations during their history. Whether they received any genetic contribution from the Far East had remained, for most of the history of Jewish genetic research, an entirely unasked question. The populations that had shaped Jewish genetics through admixture were understood to be European, North African, and Middle Eastern. The populations that Jewish communities had lived among during the long centuries of diaspora in the Mediterranean world and the Islamic world. The possibility that the genetic history of Ashkenazi Jews might extend eastward along the ancient trade routes that connected the Mediterranean world to China, across the vast steppe and mountain landscapes of Central Asia, was not a hypothesis that most researchers had taken seriously enough to investigate. A study that analyzed mitochondrial DNA variation from more than 55,000 Eurasian individuals found something that changed that picture entirely. The existence of some Eastern Eurasian haplotypes in Eastern Ashkenazi Jews supports an East Asian genetic contribution, likely from Chinese populations. This is a finding that requires a moment of careful consideration to fully appreciate. Haplotypes are specific combinations of genetic variants that are inherited together and that serve as markers of particular ancestral lineages. Eastern Eurasian haplotypes are genetic signatures characteristic of populations from East Asia, populations that conventional models of Jewish genetic history had placed on the far side of a continental divide that Jewish communities were not expected to have crossed in any genetically meaningful way. Finding those signatures in Ashkenazi Jewish genomes was not something that any existing model of Jewish genetic history had predicted, and the question it immediately raised was not merely whether the finding was real. The data was clear enough on that point, but how and when it had happened. Further evidence indicates that this connection can be attributed to a gene flow event that occurred less than 1,400 years ago, which falls within the time frame of the Silk Road scenario and fits well with historical records and archaeological discoveries. The Silk Road, the network of overland and maritime trade routes that connected China to the Mediterranean world across the vast interior of Eurasia, was at its most active and most culturally productive during precisely this period. Jewish merchants and traders were documented participants in Silk Road commerce, operating at nodes across the network from the Levant to Persia to Central Asia. There is even historical evidence of a Jewish community in China itself, in the city of Kaifeng, whose presence is documented from the period of the Song dynasty, and whose descendants maintained a distinct Jewish identity for centuries before their community eventually dissolved through intermarriage with the surrounding Chinese population. The observed genetic contribution from Chinese populations to Ashkenazi Jews demonstrates that the historical exchange between Ashkenazim and the Far East was not confined to the cultural sphere, but also extended to an exchange of genes. This sentence, in the careful and measured language of a peer-reviewed scientific publication, contains an implication that is genuinely extraordinary. The genetic connection between Ashkenazi Jews and East Asian populations is not a metaphor. It is not a cultural borrowing or an intellectual influence or a shared commercial relationship that left traces in the historical record. It is a biological fact, the physical presence in the mitochondrial DNA of Ashkenazi Jewish individuals today, of genetic variants that trace back to ancestors who came from or had biological connections to populations in East Asia approximately 1,400 years ago. The Silk Road, a trade network usually discussed in terms of silk and spices and the movement of ideas and technologies between civilizations, also moved genes, and some of those genes ended up in the people we now call Ashkenazi Jews. The mitochondrial DNA is particularly significant in this context because it is inherited exclusively through the maternal line, passed from mother to child without recombination, preserving the matrilineal ancestry intact across generations in a way that complements the patrilineal signal carried by the Y chromosome. The presence of East Asian mitochondrial haplotypes in Ashkenazi Jews means that the connection registered by the genetic data was not primarily a commercial or diplomatic relationship between Jewish men and Chinese trading partners that left no biological trace on the female side. It means that women of East Asian ancestry, or their daughters or granddaughters, entered the gene pool of ancestral Ashkenazi communities at some point during the Silk Road period. How that happened, through what specific social mechanism, and what geographic location along the vast expanse of the Silk Road network, is a question that the genetic data alone cannot answer. But that it happened is written in the DNA of living people today, in a molecular record that no subsequent historical revision or cultural forgetting can erase. The connection between these multiple lines of unexpected discovery, the First Temple period, Israelite DNA from Kiryat Yearim, the genetic kinship between Samaritans and Jewish Kohanim, the East Asian mitochondrial signal in Ashkenazi genomes, is not merely that each of them is surprising in isolation. The deeper connection is that each of them illustrates, in a different way and through a different mechanism, the same fundamental truth about what the ancient DNA revolution is doing to our understanding of Jewish history. It is not simplifying that history. It is not providing clean, reassuring answers to questions that were previously muddied by incomplete evidence or motivated interpretation. It is doing the opposite. It is revealing a past of extraordinary complexity, one in which the boundaries between populations that subsequent historical narratives drew with great sharpness and confidence were in reality considerably more permeable, more contested, and more interesting than any of the available non-genetic evidence had suggested. The Samaritan connection challenged the boundary between Israelite and non-Israelite that the biblical text had drawn with such theological force. The East Asian connection challenged the geographic boundary of the Jewish world that centuries of diaspora history had seemed to establish as self-evident. The First Temple period DNA from Kiryat Yearim challenged the gap between the ancient Israelites of the biblical narrative and the living populations that claimed descent from them, a gap that had previously been measurable only in textual and archaeological terms, and that can now be measured in molecular terms with a precision and a directness that no other form of historical evidence can match. What sits at the center of all of these unexpected connections? What makes them not merely a collection of individual, surprising findings, but a coherent and cumulative transformation of an entire field of inquiry, is the nature of the evidence that produces them. Ancient DNA does not lie in the way that other forms of historical evidence can lie. It does not reflect the political interests of the scribes who recorded it, the theological commitments of the communities that preserved it, the nationalist agendas of the states that funded the scholarship that analyzed it, or the cultural assumptions of the researchers who interpreted it. It reflects, with a fidelity that no human-generated document can match, what actually happened in the biological history of a population. It is not the only kind of evidence that matters in understanding Jewish history. Textual evidence, archaeological evidence, and the testimony of living communities all remain essential. But it is the kind of evidence that, when it disagrees with the other kinds, demands the most serious and rigorous engagement, because it is the hardest to explain away. The researchers who have produced these findings are careful, methodologically rigorous, and consistently modest in the claims they make for their data. The principal component analysis suggests a common ancestry of Samaritan and Jewish patrilineages that predates the Assyrian conquest. It does not prove a simple, unbroken descent. The East Asian mitochondrial signal in Ashkenazi DNA indicates a gene flow event during the Silk Road period. It does not reconstruct the specific individuals or communities through which that gene flow passed. The First Temple period DNA from Kiryat Yearim provides a first glimpse of the genetic history of the ancient Israelites. It does not, from two individuals in a single tomb, characterize the full genetic diversity of an entire ancient civilization. The researchers know what their data can support and what it cannot, and they are scrupulous about maintaining that distinction in their published work. But the cumulative picture that emerges from placing all of these findings in conversation with each other is one that no careful reader of the evidence can reasonably dismiss. The genetic history of the Jewish people is not the history that was previously assumed. It is older than the models suggested in some dimensions and more connected to unlikely partners in others. It includes ancestral lines that reach into the mountains between the Caspian and Black Seas, and into the trading cities of Central Asia along the Silk Road, and into the tomb of a family in a village outside Jerusalem during the First Temple period, and into the genome of a tiny community in Ateli, a Aviv suburb that has been insisting on its Israelite heritage for 2,500 years, and has now been shown, by the most rigorous and least politically motivated form of historical evidence available, to have been telling something close to the truth. Whether the people who will be most affected by these findings, the members of the Jewish communities whose ancestry is being rewritten by this evidence, the members of the Samaritan community whose genealogical claims are being validated, the descendants of the communities along the ancient Silk Road whose biological connection to Jewish history they may not have previously known, find them surprising, unsettling, illuminating, or simply irrelevant to their sense of who they are and where they belong, is a question that genetics cannot answer, because it is a question about identity and meaning rather than about molecular sequence data. What genetics can answer, and what the ancient DNA research is now answering with growing precision and growing confidence, is the factual question of what happened. And what happened, as the evidence continues to accumulate, turns out to have been considerably more surprising, considerably more far-reaching, and considerably more human in all the complicated and unpredictable senses of that word than anyone had previously understood. The connection that no one expected, not the priest's kinship with the outcasts, not the medieval merchant's descendants carrying East Asian DNA, not the first temple family buried near the Ark's resting place, turns out, on examination, to be the kind of connection that human history is always generating in the spaces between the stories that civilizations tell about themselves, the spaces where people actually lived and traded and intermarried and moved and left their biological mark on the populations that came after them. The ancient DNA is reading those spaces for the first time, and what it finds there in the molecular record of lives lived 4,000 years ago and 1,000 years ago and 2,500 years ago is a story more complex, more connected, and more surprising than the texts and the traditions and the centuries of scholarship that preceded it had ever fully managed to reveal.